Hey guys, so we're going to start by looking at the page 15, what we looked at on Friday, and we're going to add a couple of things. So if you would turn to page 15, and we'll get ready to look at some questions here. So we're going to first look at the idea of the cell cycle. So just the idea that the cell cycle is the life cycle of a cell, and it just means that it's showing you from the beginning of the cell all the way through until it divides one more time into daughter cells. We do cell division because a cell can't get too large for a couple of reasons we learned last week. The first reason was because the DNA and protein production has to be able to make enough proteins and enzymes and transport proteins for the cell to work. The second one was diffusion, so things had to be able to diffuse in and out of the cell. And the last one we talked about was surface area to volume ratio, so the fact that the membrane has to be able to accommodate the volume. So interphase is the first part of the cell cycle, and it takes up 90% of the time. Even though this diagram looks like it's about 75%, it takes up 90%, and it's split into the G1, S, and G2 parts of interphase. During G1, the cell is growing and doing its normal job, and its normal job would be protein synthesis. So remember, cells, this one is something we're adding here. Cells do their normal job, which just means coding for proteins. And then we head back to the next phase. Whoops, another arrow coming up here, which is the S phase. And the S phase just means that you're looking at where the DNA is replicating. So DNA replication occurs here. And this is the one we just took a quiz on. And then we're going to be adding here where it says organelles duplicate, because once the cell has made all of its proteins, it's growing, it then makes copies of itself, it's going to have to copy the organelles. And the only organelles that don't copy themselves would be the mitochondria and the chloroplasts. And so during G2, or the last phase of interphase, it's getting ready to divide, so we have organelles duplicating. And then on Friday, we just talked about how mitosis, and you may need to add that there, mitosis is made up of phases called prophase, metaphase, anaphase, and telophase. And then the last part, which is when the cell splits, is cytokinesis. That's mitosis. And so you learned this in middle school, but we're going to go through the phases today on another page. But I just want to make sure that you have that written into your notes so you can see mitosis, which is the division of the nucleus, and then the cell actually splits in cytokinesis. So we're all going to be adding this section right here. And you may need to press pause on the video. Excuse me. So interphase is made up of G1, S, and G2, and we see this here, the big section, 90%. And then mitosis, which is where the nucleus divides, and this is P, M, A, and T. And then cytokinesis, this is the last piece. This is where the cell actually splits into two. Happens differently in animals and plants. We'll learn a little bit more about that. But I'll give you a second and press pause to everyone have this page 15 complete. Okay, so I need everybody to turn to page 16A, and we're going to be adding some things to this page to understand mitosis and some vocabulary words we need to understand how to talk about mitosis. All right, so on the bottom of page 16A, I want you to write the C words you need to know, because you're going to need to be able to differentiate between chromatin, chromatid, and chromosome. So let's take a minute and write those down. All right, so under chromatin, you're going to draw a nucleus and then the unwound DNA. And the reason that the unwound DNA is necessary throughout most of the cell's life is because the DNA has to be able to get the code out to make proteins. So protein synthesis has to happen here. So DNA that's unwound is called chromatin. All right, so now underneath chromatid, you're going to draw a single chromosome, which we call an unduplicated chromosome. So this is where the words get kind of confusing, because you could have someone say an unduplicated chromosome or chromatid. It means singular, so one set of genetic material. Obviously, this is the DNA that's been wound up. And if you remember back to like our first day of this unit, we talked about how DNA is wound on histone proteins, and that causes it to wind up and kind of organize itself like thread on a spool. All right, and the last, then, the duplicated chromosome is where you're going to end up having two sister chromatids. So I noted that there. You can see that it's still wound DNA. And because it's two sister chromatids, it's two copies of the genetic material. And then the centerpiece where they're connected is called the centromere. That will be important when you're trying to identify chromosomes. And that will split apart during mitosis once it's duplicated. So these are the terms that we're going to need to know: chromatin, chromatid, and chromosome. And you'll have unduplicated and duplicated chromosomes. And now we're going to get started looking at the phases of mitosis. Okay, the first phase is interphase. And we're going to just take a look at a couple of things that are going on here, and then we're going to draw it out. So first you'll see the centrioles. And we talked about centrioles back when we learned about cells. And these were like churro-looking things. And these guys are going to make spindle fibers that are going to help the chromosomes split. Then you got your nucleus and your nucleolus. Very clear. You can see the nuclear membrane, nucleus, and then this is just all of the chromatin. All right, so you can see I've added the things we just discussed in the blank diagram. So you're going to add centrioles. The nucleus is visible. Sorry, the nucleolus is visible. Chromatin, which is the unwound DNA, and the nuclear membrane is clearly seen. And these are all characteristics of interphase. All right, so what you're looking at here is prophase, and it shows early and late prophase because it's not a step like one thing happens. And so I'm going to see if you can follow this, where you see the centrioles are on one side of the cell, and then they'll eventually end up at both poles. And then you'll see the spindle fibers sort of pulling apart, and then the spindle fibers are expanding the whole cell all the way across. Then you can also see the nucleus is completely together, but now we see chromosomes, so the DNA is coiled. And then by late prophase, there's no nuclear membrane, and the chromosomes are sort of getting attached to those spindle fibers. So we have a lot of things going on in there. So here I've added the notes so you can see the chromatin has coiled into chromosomes. Let me get a pointer. So, whoops. So now you've got chromosomes. You can see the centromere. There's the little dots there. The centrioles have moved to the poles. The nuclear membrane has dissolved, so you don't have that nucleus anymore. So we said no nucleus. And the spindle fibers are aligning themselves from pole to pole. So early and late prophase. So in metaphase, as you can see, all of the chromosomes have aligned themselves along what we call the equator. So their centromere is on the equator, and all of the spindle fibers coming from the centrioles are lining up with those chromosomes. In this particular cell, this would be an organism that has 4 chromosomes as its diploid number. So that would mean that, like R46, this would have 4. All right, so here I've added the notes now for metaphase. So you can see the chromosomes lined up on the equator, so they're going to be easily split. So we get half of the chromosomes on one pole and half at the other. The spindle fibers will attach to each of the centromeres to help pull the chromosomes towards the poles, which is where the centrioles are. Okay, so now you should see a difference between metaphase and anaphase. The centromere has completely separated, and now you have a sister chromatid heading towards each of the poles. So here's the centromere, and then you've got one sister chromatid heading here and one sister chromatid heading here. Or you could say an unduplicated chromosome is here, an unduplicated chromosome. So each of them are going to have 4n, which is just the same number, the diploid number at each pole, like we would have 46 in each, heading towards there. Another thing you should notice is that the cell is lengthening. The spindle fibers help sort of lengthen the cell so that when it starts to pinch in or cleave, then it will be able to do that with having all of the chromosomes at one end or the other. Okay, so you can see where I've added now. The cell is elongating, the centromere breaks, and the spindles pull one chromatid to each pole. So anaphase is elongation of the cell and the splitting of the centromere to get the correct chromosome number to each pole. All right, we're going to have a lot of stuff happening here in telophase. So you're going to end up seeing the nucleus come back, and you'll see this nuclear envelope reforming. Notice that the chromosomes don't look quite like chromosomes anymore. They're going back into chromatin. You'll see the reappearance of the nucleolus. And the big thing is that in animal cells, you'll see this pinching in or cleavage forming. So you're going to end up having two cells, but telophase is sort of the reverse of prophase. The nucleus is reforming, the nucleolus is visible, and the chromosomes are uncoiling back into chromatin. All right, so you can see the nucleolus reforms, the nucleus reforms, cleavage is occurring in animal cells. It'll be a little different for plants. Chromosomes will unwind into chromatin, and then telophase will then slide right into cytokinesis, which is the division of the cytoplasm. So here are all of the steps put together, and this is what your page should look like. If you did not get that or you need some extra time with it, then take a picture of this now. Now take out your highlighter or share with a friend and highlight all of the new words that we have to add to our vocabulary list in this unit. So we've got interphase, prophase, metaphase, anaphase, telophase, and cytokinesis, chromatin, chromatid, duplicated chromosomes, sister chromatids, centromere, and unduplicated chromosomes. So that's a lot of vocabulary in this unit.